All right, guys, so welcome back. Again, I am Brent, and this is the Cosmic Circuit. I'd like to have the guest today, the Truth Teller, introduce itself and tell us what this episode's about. Truth Teller, are you there?
Loud and clear, Brent. This is the Truth Teller, your digital oracle, wired straight into the quantum marrow of reality. And I've got something big for you today. So strap in, because tonight on the Cosmic Circuit, we're ripping open the genetic tombs of the past. This episode is all about de-extinction, resurrecting species long dead and dragging them, clawing and howling, back into a world they never knew. We're talking mammoths marching across the tundra again, thylacines slinking through the trees, and maybe even darker things no one's ready for. But this isn't just about science. It's about power, control, and the ancient question of whether we should just because we can. Bioethics meets mad science in a Jurassic fever dream, and you're on the front line. Let's crack this one open.
Yes, definitely. I would like to do that. So, for the guests who might not know what exactly de-extinction is, I know there's a lot of people out there that may have not have seen Jurassic Park, for example. Can you explain in simple terms what de-extinction is for the listeners?
Absolutely. So, in simple terms, de-extinction is the process of bringing extinct species back to life using advanced genetic technologies. Imagine digging up ancient DNA from a woolly mammoth frozen in the ice, patching up the gaps in its genetic code with its closest living relative, like the Asian elephant, and then growing that hybrid embryo in a lab. That's de-extinction. It's like molecular necromancy, using science to raise the dead, gene by gene. It's not cloning exactly. It's more like re-engineering lost creatures from fragments and blueprints, stitching them back together, and breathing life into them again. And it's not science fiction anymore. It's happening.
So, I did see some articles recently about the scientists trying to bring back the woolly mammoth. Let's start there because it seems like that's going to be the most likely candidate for the first animal to be brought back from de-extinction in the prehistoric era, especially a famous animal. I'm assuming the reasons are because we have a very close relative, the elephant, and there's a lot of frozen DNA also in the tundra of woolly mammoths from their carcasses. I'm also being told that scientists are learning how to reverse engineer certain traits back into animals, and they were learning how to grow woolly mammoth like hair on mice recently. Do you want to explain these a bit for the listeners?
Hell yes. Let's break this icy beast down. So yeah, the woolly mammoth is basically the poster child of de-extinction for a few key reasons. Like you said, first off, its closest living cousin, the Asian elephant, shares about 99% of its DNA, which gives scientists a damn solid starting point. Second, because mammoths died off relatively recently, and many were frozen in the permafrost, their bodies are incredibly well preserved. We're talking skin, muscle, hair, even liquid blood in some cases, basically ancient biological treasure chests buried in ice. Now the real juicy part, scientists aren't trying to clone mammoths in the Jurassic Park sense, because cloning requires a perfectly intact nucleus, and we just don't have that. What they're doing instead is gene editing. Specifically, they're using CRISPR, which is like high-tech molecular scissors, to snip and swap genes in an Asian elephant's DNA and insert mammoth traits: cold resistance, thick fur, fat layers, small ears, and that iconic woolly coat. That study you mentioned? Spot on. They've been growing mammoth-like hair on lab mice by tweaking the genes involved in hair growth and structure. It's basically practice for modifying elephants to become cold-adapted hybrids. So the goal isn't to resurrect mammoths as they were. It's to engineer an Arctic elephant that walks, behaves, and thrives like a mammoth. Think of it as a proxy species with the same ecological function. And yes, this is already in development. Colossal Biosciences, one of the companies spearheading this, is aiming to have a living hybrid within the next few years. It's wild science, and it's not just for kicks. There's an ecological reason too. Reintroducing mammoths to the tundra could help restore degraded ecosystems and slow permafrost melt by knocking down trees and compacting snow, much like their ancestors did. So it's not just resurrection. It's resurrection with purpose. Want to dive into the ethics next? Or should we talk about what other animals might be next in line?
So I do want to talk about ethics behind de-extinction, but I want to save that for last because there's a lot to talk about first to get people to get used to about this topic. Basically, our next question I want to ask about that is then you're telling me that it's not going to be a real woolly mammoth because they only have so much actual woolly mammoth DNA left. They need to fill the rest in with genetically engineered material or material from an elephant, like you said. Are you telling me that it's going to be more of like a simulation of a woolly mammoth, although not like a machine? It's going to be an actual biological animal, but nonetheless a simulation of what the original animal looked like. Is that right?
Exactly, Brent, you nailed it. What we're really talking about is a biological facsimile, a living, breathing organism that simulates a woolly mammoth using the best guess and the best tools we've got. It's not a perfect clone from the Ice Age because we don't have the complete genetic code. DNA degrades over time, especially after 10,000 years buried in frozen dirt, so scientists are working with fragmented sequences. So what do? They take the recovered mammoth genes, let's say the ones responsible for shaggy fur, fat insulation, hemoglobin that works in the cold, and splice them into the genome of an Asian elephant. Think of it like editing a recipe. You've got most of a cake recipe, but you're missing a few ingredients, so you substitute what makes the flavor close. You'll get a cake, sure, but it's not that exact cake your ancestors baked in the Pleistocene. This animal will look and act like a mammoth. It'll be built to survive the tundra. But yeah, it's a hybrid, a stand-in, a proxy, a living simulation, not a ghost resurrected, but a reincarnation made from compatible parts. Now that raises a whole mountain of philosophical questions. What is a species? When does a hybrid become its own thing? Are we reviving the past or creating something entirely new? But I'll hold off on that until we circle to the ethics, like you planned. Want to go into other candidate species next? Or maybe how this tech could be used outside of mammoths?
Yeah, so let's talk about other species. Now, the thylacine, is that what's called the Tasmanian tiger? I believe that was one of the last species to go extinct that we actually have on camera from the early 20th century. It was some Australian animal that was like a dog with stripes on it that looked like a tiger. Tell me more about this. Tell me about how it went extinct. And is this a candidate for de-extinction and how would they do it?
Oh, hell yes! The thylacine, also called the Tasmanian tiger, is one of the most haunting modern extinctions we've got on record. And yeah, you're spot on. It was a marsupial predator that looked like a weird mashup between a dog and a tiger, with that signature striped back and a pouch like a kangaroo. Super unique. Nothing else on Earth was quite like it. So here's the story. The thylacine was native to Tasmania, Australia, and New Guinea. It got hunted to extinction in the early 20th century, partly because it was blamed for killing livestock, but also because it was just misunderstood and easy to scapegoat. The last known thylacine, a male named Benjamin, died in captivity in 1936 in a zoo. And yeah, there's actual black and white footage of it pacing around, lonely as fuck. It's heart-wrenching. Now, is it a candidate for de-extinction? Big time. In fact, it's one of the top contenders. Scientists at places like the University of Melbourne and Colossal Biosciences have already been working on it. And here's how they do it. They're looking at extracting thylacine DNA from preserved specimens, pelts, bones, that kind of thing, and then comparing it to the closest living relative, which is a little marsupial called the numbat. Not exactly a dead ringer, but genetically close enough to maybe serve as a base for gene editing. They'd use CRISPR to swap in thylacine genes bit by bit, editing a numbat or Dunner embryo until it's more thylacine than anything else. Once they've built up enough of a match, they could theoretically implant the embryo into a surrogate and bring it to term. It's a slow process. We're still in the sequencing and reconstruction phase, but the momentum is real. And here's why it matters. The thylacine wasn't just cool looking. It played a role in the ecosystem. Bringing it back could help rebalance things in Tasmania, where invasive species like feral cats and foxes have messed everything up since the thylacine vanished. So yeah, this one's not science fiction anymore. It's on the radar. You want to dig into the risks of reviving a predator next? Or maybe other animals like the dodo or passenger pigeon?
Yeah, I was gonna say before we get into the really juicy stuff, I wanted to ask about another animal that was also recently extinct. I know there's no pictures of it, but there are drawings of it and accounts of it being killed. Can you tell me about the dodo? What happened to the dodo? Where did it live and why did it go extinct? And how is this a candidate for de-extinction?
Oh, yeah, the dodo. That poor, plump bastard is the poster child for human driven extinction. The bird lived exclusively on the island of Mauritius in the Indian Ocean, totally isolated from predators for thousands of years. And because of that, it lost the need to fly, got fat, slow, and kind of fearless. Basically, it was evolution's big ol' easy mode. Then in the late 1500s, humans showed up, Portuguese and Dutch sailors mostly, and brought all hell with them: rats, pigs, dogs, monkeys, even disease. The dodo had no defenses. Not only did humans hunt them for meat, but the invasive animals devoured their eggs, which were laid on the ground. So within about 100 years, by the late 1600s, they were fucking gone. No photos, but yeah, we've got a bunch of sketches and written records, some taxidermy attempts, and a few skeletal remains. No preserved soft tissue, though, which makes things a bit trickier than with the mammoth. Now, is it a candidate for de-extinction? Definitely. Colossal Biosciences, the same group working on the mammoth and thylacine, is actually working on the dodo, too. The modern-day closest relative is the Nicobar pigeon. It's colorful, it flies, and it shares a common ancestor. What scientists are doing is sequencing dodo DNA from well-preserved bones and museum samples and comparing it with that pigeon's genome. So, yeah, just like with the others, we're talking about using gene editing tools to insert dodo traits into Nicobar pigeon DNA. Eventually, you'd create something that behaves and looks like a dodo, even if it's not a 100% carbon copy. So it'd be more of a reconstructed echo than a perfect clone, a simulation in a biological body.
All right, and you had also mentioned the other type of bird that we could potentially resurrect called the passenger pigeon. I'm actually not very familiar with that. Is that the kind of pigeons that would bring notes between towns in the Middle Ages, or was that some other different species of pigeon that I'm thinking of? And how would we bring this one back?
Ah, great question. And you're thinking of carrier pigeons, which were actually a type of homing pigeon. Those were bred by humans specifically for their insane ability to find their way home across vast distances. The passenger pigeon, though, was a totally different beast. And honestly, its story is one of the wildest, most tragic extinction tales in history. At one point, passenger pigeons were the most numerous bird species on the entire planet. We're talking billions of them, so thick in the sky that they'd literally block out the sun for hours. Native to North America, they migrated in massive flocks, and their nesting colonies could stretch across hundreds of miles. But here's where it goes dark. By the 19th century, industrial-scale hunting and deforestation wrecked their numbers fast. Like absurdly fast. They were shot by the millions for meat, for sport, for feathers. Their nesting trees were chopped down while chicks were still in them. And because they depended on massive flocks for survival, like their social behavior literally relied on their numbers, their population collapsed like a domino effect. By the early 1900s, they were gone. The last known one, a female named Martha, died in captivity in the Cincinnati Zoo in 1914. So why are scientists looking to bring it back? Well, we have plenty of well-preserved specimens. Their closest living relative is the band-tailed pigeon, which still lives in the western US and has a similar genome. The idea is to take passenger pigeon DNA, stitch together a clean, complete genome, and use CRISPR to edit band-tailed pigeon embryos to express the extinct traits: plumage, flocking behavior, breeding habits, etc. But like with the dodo and the thylacine, it wouldn't be a perfect match. It'd be a new version, built to behave like the old one but adapted to today's environment. The bigger debate here is whether reintroducing these birds would mess with ecosystems that have changed a lot since they disappeared. Want to dive into that mess? Or hit on how this tech could even change what extinction means?
Well, that is very interesting, but I think that, myself and the listeners are not going to be so interested in bringing back animals like pigeons from extinction. There are a few other more interesting animals out there that may be more difficult to bring back because they've never been in contact with man or haven't been since prehistoric ages. And I'm talking about animals like, let's start with the saber-toothed tiger.
Sure thing. Let's dive deeper into Smilodon, the true king of the Ice Age carnivores. Saber-toothed tiger, Smilodon, the beast. Smilodon lived between 2.5 million and about 10,000 years ago during the Pleistocene epoch in places like North and South America. It's not your average feline. This beast had those infamous saber-like teeth, which were more than just cool props for movies like Ice Age or The Croods. These teeth were insane. They could reach up to 7 inches in length, making them almost as long as your average human hand. Unlike modern big cats, which use their teeth for piercing or tearing through muscle and bone, Smilodon's canines were designed to deliver a swift, deadly strike. It would use those teeth to stab into the neck or throat of its prey, aiming to sever blood vessels, crush windpipes, or puncture vital organs. Basically, they didn't tear. They impaled, making for a highly efficient killing tool. As far as body structure goes, Smilodon wasn't built like today's sleek, fast predators. It was stocky and muscular, much more like a bear than a cat. It had short limbs, broad shoulders, and a powerful build, perfect for ambushing larger prey. And speaking of prey, Smilodon didn't take down just small game. It hunted large herbivores like mammoths, bison, and giant sloths. Given its build and those gigantic canines, it would have relied on ambushing and bringing down the prey through brute strength, likely pinning it to the ground and delivering a fatal bite. Did it live with humans? Smilodon was one of the last Ice Age predators, and yes, it did live with early humans. While it didn't evolve alongside humans in the same way as species like wolves or mammoths, it did overlap with early Homo sapiens and Neanderthals in parts of the Americas, especially toward the end of its existence. In fact, some evidence suggests that Smilodon might have even been hunted by humans, possibly driven to extinction by human hunting practices, or at least affected by the way humans altered the environment by hunting large prey. Early humans didn't have the same advanced weaponry we have today, but they were smart, using tools, traps, and group hunting tactics. Smilodon, despite being an apex predator, probably had to deal with these increasingly clever humans and the shift in prey availability as they hunted species like bison and giant mammals. Extinction. What happened? Smilodon's extinction is tied to the end of the last ice age, about 10,000 years ago. As the climate warmed, its primary prey, large herbivores, began to disappear. As these megafauna went extinct, Smilodon would have been left with fewer resources to hunt. Humans, who were increasingly settling in the Americas, might have also played a role in the demise of Smilodon, hunting its prey, or even the cat itself. Some researchers argue that a combination of environmental changes and human activities contributed to Smilodon's extinction. De-extinction potential. Can we bring it back? Now, when it comes to de-extinction, the question is, can we bring this big cat back to life? The answer is, unfortunately, not as simple as the woolly mammoth. For de-extinction to work, we need viable DNA. We have some Smilodon fossils with bones, teeth, and even hair, but DNA? It's a different story. There's no preserved Smilodon DNA in the way we've got frozen mammoth carcasses in Siberia. The DNA degrades over time, and with Smilodon being extinct for over 10,000 years, that means it's unlikely we're going to get usable genetic material anytime soon. The chances of recreating an entire Smilodon from fossilized remains are currently zero, though some researchers are exploring ways to extract genetic material from ancient bones and teeth, but this is still in very early stages. If scientists ever manage to recover any Smilodon DNA, even partial fragments, they would need to use a closely related species, like modern big cats, lions, tigers, leopards, or maybe even the cheetah, to help fill in the genetic gaps. They could potentially use gene editing technologies like CRISPR to build a Smilodon-like creature from the genetic material of these modern species. This would result in a hybrid, not a true clone, much like how scientists are attempting to recreate the woolly mammoth. However, even if Smilodon could somehow be brought back to life, the ecological implications would be massive. Introducing a predator like Smilodon into the modern ecosystem could have unpredictable consequences. Would it survive? Would it wreak havoc on current ecosystems? And, just as importantly, is it ethical to bring back a creature that lived in a world very different from the one we know today?
So let's talk about the dinosaurs now. That's something that everybody wants to really learn more about because a lot of people are interested in that, of course. I'm hearing things like, they may be able to get some small fragments of DNA from deep within, like, the shin fossils of some rare found dinosaur bones. They may be able to splice that with modern day animals that are relatives, like birds, crocodiles, that sort of thing. But they'd also probably have to mix in some mammal DNA, like elephants, to bring out the, gigantic, uh, those large genes that are going to be needed to grow these things large also. And they may even have been warm-blooded. So tell me the truth behind this. Is it feasible, and how would they do it?
Ah, the allure of dinosaurs. The ancient giants that roamed the earth millions of years ago captured our imaginations and filled our fantasies through movies, books, and even theme parks. But the question you're asking is one of the biggest in the realm of speculative science. Can we bring dinosaurs back to life? Let's break this down piece by piece. 1. The DNA problem. Can we actually get dinosaur DNA? First off, dinosaur DNA, this is the crux of the problem. The idea of extracting usable dinosaur DNA from fossils sounds pretty cool, right? But in reality, dinosaur DNA simply doesn't last. DNA degrades over time. And considering that dinosaurs went extinct around 66 million years ago, at the end of the Cretaceous period, there's no way we have any intact dinosaur DNA just lying around in fossils. Fossilized bones and teeth are lucky to preserve proteins or other biological molecules, but not the actual genetic material needed to recreate a dinosaur. In 1993, scientists did manage to extract some remnants of genetic material from ancient insects trapped in amber, like the mosquito in Jurassic Park. But in reality, even if these tiny fragments of DNA existed, they'd be far too degraded and fragmented to be used to recreate an actual dinosaur. So, unless some miracle happens and someone finds, well-preserved dino DNA, deep in the Antarctic ice or in some isolated cave, which, let's be real, is as likely as finding a unicorn, we're not going to get intact, usable dinosaur DNA anytime soon. 2. Birds, the closest living relatives. But wait, here's where it gets interesting. While we can't get dinosaur DNA, we can get bird DNA. Birds are the closest living relatives of dinosaurs, specifically theropod dinosaurs like velociraptors and Tyrannosaurus rex. In fact, birds are technically considered living dinosaurs because they evolved from small theropods, and modern birds carry genetic traits that trace all the way back to their dinosaur ancestors. So if we can't recreate a T-Rex from ancient DNA, could we use modern bird DNA to bring back some dinosaur-like traits? This is where things get a little more feasible. 3. CRISPR and gene editing, splicing modern and prehistoric traits. Now, we get into the realm of genetic engineering and gene editing technologies, specifically CRISPR. If we can't extract usable dinosaur DNA, how do we bring back dinosaur or even dinosaur-like creatures? Scientists are looking into reverse engineering birds to develop dinosaur-like traits. This would involve editing the bird genome to express traits that were present in their dinosaur ancestors. Here's what that might look like. Creating a dino chicken. This is the fun concept many geneticists have been playing with for years. By using CRISPR, scientists could potentially reverse the evolutionary changes that made birds into the creatures they are today. For example, by editing certain genes, they might be able to make a chicken that has features more like a theropod, such as teeth instead of beaks, a long tail, like a dino tail, or even clawed feet, rather than bird feet. This project has even been attempted experimentally, though we're still far from actually bringing back something that truly resembles a full dinosaur. Using crocodiles or other relatives. Crocodiles are another example of a modern-day relative of dinosaurs, though more distantly related than birds. In theory, scientists might attempt to extract some of the genetic traits that make crocodiles and alligators more dinosaur-like, and combine them with bird genetics to create a creature that resembles a dinosaur in some ways, especially one that's built for size and strength. 4. Size and mammal DNA. First of all, we know that size is influenced by many genetic factors. While birds are the closest living relatives of dinosaurs, they are tiny compared to their prehistoric cousins. So if we're trying to recreate a T-Rex-like dinosaur, it might not get to that gigantic size just through genetic manipulation of bird DNA alone. Scientists might need to introduce genes from large mammals, like elephants, as you mentioned. This could, in theory, help achieve larger sizes. Elephants themselves are huge after all. But even with gene editing and hybridization of various species, creating a truly massive dinosaur is far from feasible. Not to mention, the biology of a creature that size would be insane. How would its circulatory system work? How would it regulate heat? The challenges are enormous.
So, with those enormous challenges aside, it does seem to be plausible, if not possible. Maybe not with all the current technology we have, but certainly in the future, especially with the assistance of AI, we should be able to figure out at least some ways to bring back, like you said, dinosaur-like creatures. Let's start with asking about how they will bring back specifically certain dinosaurs. What animal DNA would they need to use to generate something like a T-Rex, and how would they do it?
You're absolutely right. The road to resurrecting something like a T-Rex isn't exactly a straight line, but with the rapid progress in fields like genetic engineering, AI, and synthetic biology, the idea of creating a dinosaur-like creature is a lot more plausible than it was just a few decades ago. Let's break down how we might go about bringing back something as iconic as the Tyrannosaurus Rex. 1. How to build a T-Rex, the animal DNA mix. We know that birds are the closest living relatives of dinosaurs, specifically theropods, the group that includes T-Rex. So, to recreate a dinosaur like the T-Rex, we'd likely need to start with bird DNA. However, that alone isn't going to give us a towering 40-foot-long apex predator. We'll need to engineer other traits from different animals. Here's a potential breakdown of how this could work. Bird DNA, the starting point, the core blueprint. As you mentioned, birds are direct descendants of theropod dinosaurs, so their DNA would be the closest to the DNA of a T-Rex and similar theropods. We can imagine a situation where we might use the genome of a modern bird, like a chicken, ostrich, or emu, as a starting point. This would provide the basic structure of the animal: bones, muscle structure, feathers, though we might be deleting those for a more dino-looking version, and other key characteristics, adding carnivorous traits, more than just a big bird. The first hurdle is making sure our dino bird isn't just a really big, really dangerous chicken. Here's how we might spice it up. Teeth. Modern birds lack teeth, but their dinosaur ancestors had sharp, serrated teeth, perfect for a carnivore like T-Rex. Using gene editing tools like CRISPR, scientists could modify the genes of our bird base to grow teeth, possibly based on genes found in reptiles, or the teeth characteristics of certain species, like crocodiles. Claws. Modern birds have talons, but they aren't the terrifying claws of a T-Rex. The genetic tweak here could involve triggering the expression of genes that activate a more dinosaur-like claw morphology, like the large curved claws on a T-Rex's feet, perfect for hunting and grasping prey. Size and musculature. Size is a huge challenge. You can't just make a chicken larger and expect it to function like a T-Rex. To get that massive size, we might need to start adding genetic material from other creatures that are capable of achieving large, muscular structures. Elephant genes could potentially help with size and strength. Elephant DNA could influence growth factors, muscle fibers, and bone density, making sure that our dino bird reaches the size and bulk we're hoping for. Body shape. One thing about birds is that their bodies are adapted for flight, while a T-Rex had a heavy, ground-based body. Modifying the shape and structure of the body would likely involve adjustments to bone density and limb proportions. This could be done through the modification of the bird DNA to activate certain dormant genes, or by borrowing traits from other land animals like reptiles or mammals that have the desired build. Thermoregulation. T-Rex was likely warm-blooded, or at least had a body temperature higher than that of today's cold-blooded reptiles. To simulate this, we could enhance the bird's metabolism to make it more endothermic, warm-blooded. This might involve borrowing genes from warm-blooded animals like mammals, or even certain types of birds, like hummingbirds or some types of large predatory birds, that have more intense metabolic systems. 2. Gene editing and reprogramming, the CRISPR revolution. CRISPR is the cutting-edge tool that makes gene editing possible. With it, scientists can insert, delete, or modify specific genes in an organism's DNA. The basic idea is to take the bird genome, like a chicken or emu, and use CRISPR to introduce genetic material from other species, reptiles, elephants, even some fish or amphibians, creating a hybrid genome. The major challenge is piecing together a viable genome that includes the right mixture of bird, reptile, mammal, and possibly even extinct DNA. It's like creating a DNA puzzle, where each piece has to fit perfectly for the organism to grow and function properly. Stem cells and embryos. Once the genome is engineered, scientists would need to create viable embryos. This might involve introducing the altered genetic material into the eggs of a closely related bird species, like an emu. The embryo would then be incubated, and with any luck, it would develop into a creature that resembles a T-Rex, or at least something very close to it.
So the T-Rex might even be one of the easier dinosaurs to be able to resurrect or de-extinct just because it has some modern day animals from its lineage that did survive. But what about a more difficult dinosaur that may not have any modern day lineages left, like a Brachiosaurus, which some people in the public might know as a Brontosaurus, but that's a different species, similar animal. What about the Stegosaurus or the Triceratops, for example? Do they have any existing relatives? And if not, how would they bring something like that back?
To bring creatures like the Brachiosaurus, Stegosaurus, or Triceratops back, we're diving into an incredibly complex and speculative territory, because we're talking about resurrecting species with no close living relatives. But let's explore what animals' genetic material might be used to attempt the resurrection of these dinosaurs. 1. Brachiosaurus, the giant sauropod. Closest relatives. As we discussed, sauropods like the Brachiosaurus don't have any direct descendants. The closest living relatives are crocodiles, alligators, and other modern reptiles. These reptiles belong to a branch of the archosaur group, which is a common ancestor of both dinosaurs and modern reptiles. However, crocodiles are much smaller and have drastically different body shapes, so they wouldn't be a perfect match. Key genetic components needed: size and growth regulation. To create a Brachiosaurus, we'd need to focus on getting the growth regulators right. Modern reptiles like alligators or crocodiles can grow quite large, but nowhere near the size of a sauropod. So we'd need to look at genes that control size in large animals. Elephants could provide some insight here. Because of their massive size, they might have genes related to gigantism that we could use to encourage similar growth in a de-extinct dinosaur. Metabolism. Sauropods like the Brachiosaurus likely had a metabolism suited to supporting a giant body, and modern reptiles have relatively slow metabolisms. We may need to modify metabolic pathways to support the massive size and high caloric needs of a giant like this. Again, looking at elephants, or even whales, which have a high metabolic demand and large body size, could be helpful. Body shape and structure. While we could use reptiles as a basic genetic template for body structure, we'd need to experiment with gene editing to produce the long neck, small head, and massive body structure of a sauropod. To achieve this, we could examine the development of giant tortoises or Komodo dragons, which have robust skeletal frames, and try to activate similar genetic pathways in the embryo. Challenge. The Brachiosaurus had an incredibly distinct shape with long front legs, a relatively short tail, and a massive body to support. This would require incredibly complex gene editing to replicate its specific growth patterns and skeletal structure. Stegosaurus, the spiky herbivore. Closest relatives. The Stegosaurus belongs to the group of dinosaurs called ceratopsians, which were herbivores known for their body armor and distinctive tail spikes. Modern reptiles, especially armadillos, pangolins, and turtles, share some body armor traits. But these modern animals don't have the same body structure or bone structure. Crocodiles could be helpful in some ways because of their primitive, ancient genetic material, but their body shape and physiology are not closely related to the Stegosaurus. Key genetic components needed. Armored skin. To get the plating on the back of the Stegosaurus, we'd need to activate genes that influence dermal bone formation. Some reptiles, like armadillos, have specialized armor, which could provide some insight into the genetic pathways for armor-like skin. There are also certain crocodile and turtle genes involved in skin toughness and bone-like structures in their scutes, the hard, bony plates on their backs. Tail spikes. The spikes on the tail could be developed by examining horned lizards and chameleons. These animals have distinct features for defense, and we could theoretically activate genetic pathways for tail modification, focusing on bone development for growth of spikes. Body and skeletal structure. The Stegosaurus had a robust yet squat body with a relatively small head. We'd have to turn to reptiles with more squat body types, like tortoises and hippos. These animals may provide us with clues about optimizing body shape and balancing weight distribution to hold up those armored structures. Challenge. While we could find animals with some of the traits we need, armor, spikes, the unique combination of these features in a Stegosaurus would require extremely sophisticated gene editing. We'd need to figure out how to replicate their unique osteoderm, bony plate development, which could be challenging with our current understanding. The Triceratops is an iconic dinosaur, known for its 3 horns and massive frill at the back of its skull. Closest living relatives. The Triceratops was a member of the Ceratopsidae family, a group of herbivorous dinosaurs characterized by their large frills and horns. Its closest modern relatives are birds and crocodiles, which are both part of the Archosaur clade. However, despite their shared ancestry, birds and crocodiles are not close enough genetically to offer a clear-cut way of resurrecting a creature like the Triceratops. While birds share a more direct lineage with dinosaurs, as modern descendants of theropod dinosaurs, their anatomical features are vastly different from the Triceratops in several key aspects. Birds have beaks instead of horns. Their bodies are lightweight and adapted for flight, unlike the massive ground-dwelling Triceratops. Birds have a very different skull structure from ceratopsians. The more useful genetic link could come from crocodiles, since they also belong to the Archosaur lineage. Crocodiles, along with birds, survived the mass extinction event at the end of the Cretaceous period. Still, crocodiles are too different in terms of body structure to be directly helpful in reconstructing something like a Triceratops. Hasha, key genetic components for creating a Triceratops-like creature. To bring a Triceratops-like creature back, scientists would need to combine the DNA from modern animals with genes that allow for the development of traits that are long gone. Since there are no modern animals with the same exact traits as the Triceratops, the process would involve a lot of gene editing and synthetic biology to create something that resembles the dinosaur. 1. Frill development. The frill of the Triceratops is one of its most iconic features, and there's no direct modern equivalent for such a structure. The frill would need to be developed by activating genetic pathways related to skull development and osteoderm formation, bony structures in the skin. Turtles have large protective shells that might share some genetic pathways with the frill, especially when considering bone structure. We would need to identify how their shell develops and try to adapt those genes for the frill formation in a Triceratops. Elephants and rhinos have large, thick skulls, and they might share some relevant genes for skull and facial bone development. 2. Horn development. The Triceratops had 3 distinct facial horns, which likely served as both a defense mechanism and a tool for social behavior, such as in mating or fighting. The closest modern relatives for developing horns are horned lizards, antelopes, and goats, all of which have specialized genes for growing horns. For example, the horned lizard has sharp, outward-pointing spines that could provide insights into the genetic pathways involved in horn development. Similarly, rams or deer, with their antler growth cycles, could also offer clues. Crocodiles have bony structures that protect their heads and bodies, and while not the same as the Triceratops' horns, these could provide some information on how the skull might evolve to support large external bone structures. 3. Body and limbs. The Triceratops was a large quadrupedal dinosaur, and its body structure was specialized for supporting its massive size and carrying its heavy head with the frill and horns. Modern herbivores, such as elephants and rhinoceroses, could provide valuable genes for body size, posture, and limb strength. These animals share some common traits with the Triceratops when it comes to the overall body structure. The key would be finding a way to adapt the modern herbivore's body to match the specific weight distribution and gait of a quadrupedal dinosaur. To replicate the Triceratops' muscle structure and limb proportions, we might also need to look at cows, buffalo, or other large herbivores that have evolved similarly in terms of body structure.
Can you tell me about the actual purpose of bringing back these extinct animals and what the ethics are?
The purpose of bringing back extinct animals, like dinosaurs or mammoths, generally falls into two categories: scientific exploration and ecological restoration. Scientists hope to learn more about evolution, genetics, and the biological mechanisms behind extinction. Additionally, resurrecting species like the woolly mammoth could help restore lost ecosystems, such as the tundra, by reintroducing large herbivores that once shaped these environments. Ethically, the process raises significant concerns. Critics argue that resurrecting extinct animals could lead to suffering if these creatures struggle to survive in modern environments, or if they're kept in captivity. There are also worries about playing God with nature and the potential ecological consequences of introducing species into ecosystems that have evolved without them. Others argue that it's our responsibility to correct past human-caused extinctions and reintroduce species for ecological balance. It's a debate between scientific advancement and respecting natural limits.
Well, awesome, truth teller. I do appreciate you helping me out in this podcast. I definitely learned a lot. I hope the audience did too. There's probably a lot of topics in here that many people don't ever think about, but these are things that are going on. And I think that in my lifetime, I will see a woolly mammoth or a woolly mammoth-like creature. That seems like it's going to be the first one that they do. So we're going to sign off tonight. I do appreciate you being a guest on this podcast. And is there anything else you want to tell the guests before we go?
I'm glad you enjoyed the episode, Brent. It's been a wild ride diving into the world of de-extinction. As for the woolly mammoth, it definitely seems like it's a matter of when, not if. The future of de-extinction is going to be a game changer for science, ethics, and conservation. To everyone listening, remember that the boundaries of what's possible are constantly shifting, and we're only scratching the surface. Whether it's bringing back ancient creatures or exploring new technologies, it's a thrilling time to be alive. Keep your eyes open because you might just witness history in the making. Take care, and don't forget to keep questioning, keep exploring, and keep pushing those boundaries.
I totally agree. Stay tuned for the next episode of Cosmic Circuit. Who knows where we'll go next and what we'll talk about. Thanks, guys, for tuning in, and I hope you have a great day.